Ever wonder what makes the greats great? What makes the successful successful? What makes the brilliant brilliant? Our Tuesday meetups with the celebrities of pharma industry and science are your one-stop shop to all these answers and more. Join us for Pies of Life, an initiative of the Biopatrika Industry Mentorship Program, where we bring your dream mentors to you.
Welcome all to our Pies of Life. This Tuesday, we are honored to welcome the man who has successfully traversed Whitaker's kingdoms. From a plant biologist to the assistant director of the Purdue Institute of Inflammation, Immunology, and Infectious Disease. Oh, yes, that is quite a leap. He's also the vice chair of the steering committee at the Big Ten Cancer Research Consortium Foundation. And if that wasn't enough, Professor Soar spends his leisure hours building the COVID-19 diagnostic sensor with his company, IdentifySensors Biologics, to help humankind even more. Now, that is not a person we meet every day. Welcome to Atete with Thomas Soares. Tommy, I'd like to invite you to tell us your life story in your words, and you can share your screen if you like.
Great. Thank you so much. And it's a pleasure to have the opportunity to speak to you and have the honor of being invited to this wonderful session. I'll try to go fairly quickly through my initial stages and not bore you too much with too many details, but there's a lot of good juicy stuff there, good science that I'm excited to tell you about, and hopefully you can be inspired. One thing that I will say is, like many of us scientists, I've been inspired and very much moved by the people I've interacted with in my life. So I'm very much like Ulysses' poem says, I'm a part of all that I have met. And really, these people have been my mentors and have inspired me to keep going and moving forward. So my venture into plant sciences really started one day when I was with a friend and we saw a firefly go by, and I said, oh, wouldn't it be cool if we could transfer the firefly genes into a plant and make it glow in the dark? And that inspired me to really start learning about molecular biology, about plant biology, plant physiology, and try to pursue this idea of genetic engineering of plants. And at first, when I got the idea, I thought, okay, well, I have to go start talking to plant biologists. And I had the opportunity of working with Dr. Roger Horton at the University of Guelph in Canada. Guelph is a city about 45 minutes west of Toronto, Canada. And that gave me the opportunity of doing this experiment where we start looking at the transpiration rate of barley leaves that were exposed to gibberellins. Gibberellins, if you didn't know, are these plant hormones that causes the shoot elongation to occur. And we were looking at how much water would be transpired with different concentrations of the gibberellins. And that was a very interesting experience. I learned a lot from that, especially how little I knew about statistics and how embarrassed you can be if you really don't know your statistics and you're talking to other scientists about the experiments that you're doing. So it was really early in my career that it said to me, oh, you really need to be prepared and you need to work hard in order to handle the different situations and be able to defend what you did. So when I pursued a master's degree, I ended up working with this professor named Stephen Bowley, who is an alfalfa breeder. And alfalfa is an obligate outcrosser. It's an allotetraploid. And to breed a tetraploid, you really need to know your statistics. You really need to know your genetics. And I learned a lot from this man in that respect. But I had the opportunity of keeping to pursue this, this passion for creating transgenic plants. And I got the opportunity of working with transgenic alfalfa, one of the largest hay crops in North America. That alfalfa grown in Canada is exposed to a lot of cold temperatures, and winter hardiness was something that we were trying to improve in the plant. And so I started working with transgenic alfalfa expressing a pyrophosphate-dependent phosphofructokinase enzyme from this parasite, Giardia lamblia. And the reason was that the phosphofructose kinase, this pyrophosphate-dependent phosphofructokinase from Giardia, was bidirectional. And we thought that that would give the plant a metabolic flexibility to take and remobilize starches and more complex sugars and mobilize them into more soluble sugars for regrowth and also for protection during the winter months. So that really, again, exposed me a lot to research, to plant metabolism, and I wanted to continue to pursue this. So I started doing this work at Purdue University for my PhD under the guidance of Jeff Bolenek in the Department of Agronomy. And what I was trying to do was really understand a particular promoter element that was controlling the vegetative storage protein genes in alfalfa. You see, alfalfa is a perenniating plant. And so when winter comes, the shoot die off and the root itself has to maintain throughout the winter to then survive and allow for spring regrowth to occur. And a lot of the reserves for spring regrowth are stored in the root in the fall and are remobilized in the springtime to allow for shoots to regrow before the plant is able to do any photosynthesis. And so there's these vegetative storage proteins that allow alfalfa to hydrolyze these proteins, break them down, and feed amino acids to the regrowing spring shoots. And so that was, again, a lot of physiology, a lot of metabolism. And I brought this element of molecular biology to the lab from my master's degree. But I saw that things were kind of moving a little bit slow, and I was more ambitious than that, and I wanted to get into a lab that was really publishing and was really cranking out papers. And I was exposed to this lab. So here's where I was talking about the remobilization of these particular vegetative storage proteins and how they were controlled by these particular promoter elements. But being more ambitious, I wanted to pursue more action. I wanted to learn more, and I was excited about this lab that was at Purdue in the horticulture department, where we were looking at particular metal accumulating plants. And one of the projects that Professor David Salt, who was my PhD advisor, was working on was looking at how particular plant species accumulate selenium from the soil. And selenium is an element that is very similar to sulfur. And so you can feed selenium to any plant that accumulates sulfur and it will choke itself. It will accumulate it as if it were sulfur. And the reason is that selenium exists very much like sulfur as these common anions, as you see sulfide or sulfate, and in the environment, you typically see selenite, selenate as well. And so the plant confuses these two and ends up making proteins with selenite and selenium instead of sulfide. And so these disulfide bridges now become diselenide bridges. And so the protein it doesn't have the right conformation, doesn't have the right shape. And so it doesn't function properly. And the plant ends up choking itself to death. But there are these particular plant species called astragalus that know the difference between what's sulfur and what's selenium and are able to compartmentalize that. And that was essentially my PhD, and how these plants are able to distinguish one from the other. And through my studies, we ended up looking at this selenocysteine methyltransferase enzyme, which takes cysteine or selenocysteine and methylates it using S-methylmethionine as a methyl donor to make methylcysteine or methylselenocysteine. And we thought that this SMT enzyme played a crucial role in allowing the plant to tolerate higher levels of selenium inside its tissues by diverting the seleno amino acids from protein production. And essentially, we came up with this pathway where the plant is able to take selenate and sulfate assimilating pathways and are able to move them out, the selenium containing amino acids, out of the pathway and allow the plant to continue to make its proper enzymes, its proper proteins. During that PhD, I had an awesome opportunity. Professor Salt, David Salt, ended up getting a grant from the government, from NSF, where he said that he would create a museum exhibit about omics and about all of the omics that we were dealing with, that we were learning, that we were exploring, that we were developing in his lab. Uh, David Salt ended up becoming the father of what's called ionomics, which is the elemental composition of organisms, and looking at how genes drive that elemental composition all the way down to the common atoms that we know. And so this omics world exhibit is something that I ended up taking, again, ownership. I saw that opportunity. I loved working on communication and educating kids about plant biology, about what we were doing, molecular biology. And I took this as an opportunity. And I said to David, I said, I'm going to do this. We're going to do a whole museum exhibit, and I'm going to include this as a chapter of my thesis. Are you okay with that? And we went and we spoke to the rest of the committee and asked them permission, and they thought it was great. So at the end, I ended up building a museum exhibit and including that as a chapter in my thesis. And the museum exhibit was called Genomics Explorer. And what I ended up doing is building a three-dimensional module of a cell where people actually walked into the cell and they were able to look at the different organelles and look at videos of the organelles. And the videos were hosted by this animated cartoon creature that I called Saito, who was your helper inside the cell and explained how the mitochondria worked or how chloroplasts work or the nucleus and what's happening. And in one of those, I ended up writing a grant to develop these video games that went along with the museum exhibit. And I can probably pop them up here at some point in time and you can guys see them. But they're interactive video games where you have to figure out the right elements to make a plant thrive. And then you can inspect when the plant is thriving or when it's not doing so well. You can inspect what's happening inside of the chloroplast, inside of the thylakoids, inside of the mitochondria, and look at what's happening with electron transport, or look at the nucleus and look at what's happening with gene expression patterns. And we submitted these three modules to a competition, and we won first place in the NSF Science journal. We were published in it, as well as first place in digital. I think it was digital platforms or something like that. And we ended up calling this the Genomics Digital Lab, which continues today and was formed even into an educational company that serves, even still today, you can access it. And it grew to be other elements, other modules that are interactive and allow you to learn as you play along. That was the idea. I was always motivated to teach and get people to learn by having fun and not realize that they were learning. During my postdoc, I had the opportunity of working with Dr. Clint Chappell, who was the head of the biochemistry department at that time. Clint is an expert in the phenylpropanoid pathway, which is the pathway in plants that leads to what are called lignin precursors. And lignin is what makes plants woody. It's the woody tissue. And what I was doing with Clint was trying to create enzyme chimeras. We were taking these cytochrome P450 enzymes that participate in the phenylpropanoid pathway, and we were taking them from different plant species, and I was trying to stitch together enzymes that were bifunctional or multifunctional by putting together these different elements from the different species. And so using a scheme of primer extension PCR, I designed and developed a robotic system by which to create hundreds and thousands of these chimeras. And start using my knowledge and my abilities in mass spectrometry also to ascertain their activity. So trying to combine both of these to try to design and synthesize enzymes that were functional. That put me in what's called the Binley Bioscience Center at Purdue University. And Purdue is, made up of colleges, and inside the College of Science, for example, you have the Department of Biology or Math or whatever it might be. But there is also this place at Purdue called Discovery Park, which does not belong to any of the colleges, and its sole purpose is just for research. And that's what I was doing during my postdoc, was trying to create these enzyme chimeras, but it exposed me to all of the realm and possibilities that are in the Binley Bioscience Center. And it put me to work with Richard Kuhn and Charles Bach. Richard was the director of the Binley Bioscience Center, and Charles was the managing director or director of operations. And I found this position as a center project manager that became open in the Binley Bioscience Center. And I applied for it. I thought, oh, okay, but I have nothing to lose. I'm doing this postdoc. I like doing this postdoc, but this project management for the center for all of the Binley sounded really interesting. And I thought, well, I don't have much project management experience formalized, but I do manage my own projects. I do manage my own experiments, and I plan them and so on. And at the end, I stuck my neck out. I asked some questions, and I was able to apply for the job, and I got the job. And that gave me the opportunity of working the center that is, I want you to think about it as a shopping mall for researchers and scientists. It is a place where you can do microscopy and look at all kinds of different cell types. So super resolution microscopy and all the way to in vivo imaging techniques as well. There's biophysical analysis. There is computational analysis as well, and informatics that goes along and helps with all of the metabolite profiling analysis that is done using mass spectrometry, or the protein composite analysis, proteomics and metabolomics. There's also a facility that is in partnership with our vet school that we call the Translational Pharmacology Facility, where we're able to do pharmacological testing in preclinical models. And so when you can imagine, you can collect samples and blood from those models, bring them back to the lab, ascertain, their metabolism, ascertain their immune response by looking at their different cell types using flow cytometry and cell separation techniques. And so this place really is a technology-driven place that serves the faculty, and faculty from all different departments. This is a neutral place where we do things in bacteria, plants, humans, as you heard me say, but mice, rats, pigs, whatever organism or model that we're working on to look at diseases, to look at explore basic biology, make new materials, and so there's a wealth of activity and dynamic movement in the Binley Bioscience Center. And that one of the things that I got to do in there is to start working in what's called the Indiana Clinical and Translational Sciences Institute. My charge as a center project manager in the Binley Bioscience Center also gave me this charge to act as what's called the navigator for Purdue in this Indiana CTSI. And the navigator is the person in that particular institution that helps connect outside of the institution, and for those outside to connect inside of the institution. The Indiana CTSI is a collaboration between Indiana University School of Medicine and also Indiana University, the Bloomington campus. There's also branch campuses that are now in cities like Evansville and Terre Haute. It's also a collaboration, and part of that infrastructure is with the University of Notre Dame in South Bend, Indiana, and obviously Purdue University. So it's three institutions put together, allied to improve translational sciences for the state of Indiana, and by which to improve the health of people in the state of Indiana. And this gave me, as you can imagine, an unbelievable network by which to work with both people in the Indiana University system and people in the Notre Dame system. And what we do is that we try to improve the efficiency by which translational science research occurs. Let's say there's an investigator at Purdue and they're doing some work on a diagnostic, like what we're going to talk about, a COVID diagnostic, and they need some clinical samples from people that are infected with COVID. So I am the one that goes and finds the collaborator at the School of Medicine who's seeing patients who can have the potential of collecting those samples that can then be analyzed with the partner at Purdue University. So these are the types of programs and ways that we try to improve how the discovery from the laboratory setting can be applied to the bedside of the patient in a little bit more concerted manner. The opportunity of the Indiana CTSI also is larger, meaning that Purdue University is a land grant institution, and essentially we have our representatives in all 92 counties of Indiana. And that allows us deep access into the populations, into the communities of Indiana. And Indiana, I'm stuck here in Purdue University between a cornfield and a soybean field. It's a very rural state, other than the urbanized centers like Indianapolis, Fort Wayne. You could say Lafayette, Greater Lafayette would be an urbanized center. The state is by far a rural state. Being part of the Indiana CTSI has also allowed me to create programs, and one of them that I'm proud to tell you about is this postdoc challenge, where postdocs get to participate in writing applications to use up to $5,000 to use some of these core facilities that I showed you in the Binley Bioscience Center. But what's more important is that I get them to participate with me in the review session following NIH guidelines. And so they get to see both sides of the coin, meaning that they get to be the applicant, but they also get to be the reviewers and see what happens behind the scenes to determine which ones are the best proposals, which one's the best science to invest our $5,000 in. So that gives them a really great perspective. It gives me a lot of joy to be able to set up programs like this and be part of that training for such an important group of people, of scientists that sometimes get disregarded. I'll just say that. Very quickly, when Richard Kuhn was asked to form the Purdue Institute of Inflammation, Immunology, and Infectious Disease in 2016, he called me. He said, Tommy, they've asked me to do this, and I would like you to come and help me do this. We had worked together for many years in the Binley Bioscience Center, and I said, absolutely, Richard, let's go put this thing together. And in 2016, we start working to form this institute that is divided into four research foci areas, one being imaging and diagnostics. We have a very strong group of engineers, biomedical engineers, and even from our human health sciences college, a wonderful group of investigators looking at MRI, magnetic resonance imaging, for example. But with that, all kinds of imaging modalities, all kinds of imaging techniques that are being used to create new types of diagnostics. We also have a division of immunology and inflammation, as you can imagine from our name, and infectious disease as well. Dr. Suresh Mittal, distinguished professor in veterinary medicine, and he's been working on adenovirus vectors for a long time, and he's working also in this area with us. We have an area of prevention and intervention as well. And I'm so glad to be able to work with such a wonderful group of people. Richard and I work closely with Robin Benson and Melanie Lindsay, who are part of our administrative team. And we serve almost as a new body for research for many faculty. They have their own departments and they have their own cultures in their own departments. But what we try to do is bring new faculty together, bring new disciplines together to go after more complex funding opportunities as well. We've done a lot of global initiatives. One thing that we were part of is bringing cryo-electron microscopy to India. Richard was part of the group that celebrated the inauguration of the first cryo-EM at INSTEM at the NCBS, the National Center for Biological Sciences in Bangalore. And we continue to work very heavily in that area of cryo-electron microscopy, especially for structural biology, structural virology, understanding the structure and function of proteins, and in that capacity, it's a remarkable piece of technology, and we formed now a consortium, a regional consortium here for cryo-electron microscopy. One of the things that I'm developing now and that you probably, I hope you get to see, I'm publishing a paper We're hopefully going to get it out very soon. I'm waiting to hear back from the journals. Is this paper on a program concept that we've been working on, which is called reciprocal innovation. Most people, when they think about global health, they think about a high income earning country going to a low income earning country and going to implement some kind of intervention there, some kind of resolution. And it seems very much that when you say global health to people, they think of this unidirectional flow of resources, information, knowledge. And I'm trying to dispel that. Where we're doing in the Indiana CTSI and the Indiana University Global Health Research Program is try to create this concept where there's a bi-directional iterative exchange of technology and methodology or processes that can help both countries. So for example, in Indiana, we have an HIV problem in southern part of the state due to needle sharing. We have an opioid heroin problem in the southern part of the state, and people are sharing needles and passing HIV along with that. In Kenya, there's an HIV problem due to unprotected sexual intercourse, but the nature, the rurality of how the distances by which people who are living with HIV and AIDS in Kenya looks very much like the distances that the people would have to travel in southern Indiana. And we've learned through our international programs, this AMPATH program that IU School of Medicine has put together, they've learned how to deal with antiretrovirals and dealing with taking care of these patients in Kenya. And they use that information now to implement similar programs in Indiana, southern Indiana. So it's this concept where we can both have mutual benefit.
So Charu, maybe you can, or whoever is coordinating the questions.
Yeah. So, I mean, till now it's been such a roller coaster. It would put Disney to shame. And I mean, you followed your passion. You made the best of every opportunity, and it really reaffirms that it doesn't matter where you start. I'm sure there's many questions. I think you can all raise your hands, and then Vikram will kind of direct you towards who gets to ask first. And if you're shy, just drop your questions in the chat box. While everybody's thinking, I already have a question. So what I wanted to ask you is that the animal and plant cells are very different. And you're now working on the animal aspect of it. So is there, do you feel that your expertise in plant or the plant-based molecular science that you study gives you more insight in some matters? And also, does it help you be a more ethical scientist?
Oh, good question. I don't know. I think it does make me be a more ethical scientist because I can't anymore take thousands of seeds and like spread them and ask my scientific question. But now if you're dealing with animals, there's a lot more sensitivity there. And so I don't want to waste life and I don't want to, cause suffering. One of the things my mom once asked me, Tommy, why are you going into plant biology? I always thought you liked animals. Maybe you should go into vet school or animal science. I said, Mom, plants don't have faces and they don't cry. So when I mash them up and I try to extract their DNA, and she kind of like, okay, but it's definitely a good question because it does make you pause, make you think. I always said when we were at the Binley Bioscience Center and we were running samples from cancer patients or something like that, I would always say, these are patients. These samples are unbelievably precious. People had to donate of their lives to give us these samples so that we could look at them under the microscope or in the mass spec or whatever we were doing. So let's treat them with a lot of care. I don't think I was that careful when I was working with plants, meaning that you know, there's more plants to be gotten, meaning. And then the other question, you know, there's a certain plasticity, if you want, between cells that a plasticity in my mind about, you know, an animal cell and a plant cell. Yes, they're very different. They function very differently. But there was a lot that I learned about the manipulation of metabolism in plants that gave me this ability to look maybe even at the DNA and the molecules that now I can relate to investigators that are doing that more, right? And so I don't know that it gives me an advantage, but I can certainly understand when they tell me, hey, you know, I'm trying to look at NADH, NADPH, I know where they're talking about. And that's universal no matter where if it's an animal cell or a plant cell. Sure, if we're talking about some photosynthetically driven activity, yeah, okay, you know, can't do that in the humans, but you know, that's obvious right there.
Shruti, please go ahead.
Hi, sir. I'm Shruti. I am from food biotechnology background, and I worked in a biologicals company for 1 year. While I was browsing through your startup company about the IdentifySensors, I could see that you develop sensors to detect chemicals. I mean, that are harmful for honeybee population. That was very fascinating because I've seen a lot of technologies using sensors for detecting, decay in food. All that those things I've seen, but this is something very different that I could see. Can you elaborate more on that?
Sure. And I want you to know that there is this company called IdentifySensors that was started before I started with them, this new startup called IdentifySensors Biologics. Okay. And so they had formed this company with these sensors for detecting not only these harmful substances, but they for herbicides, for pesticides, for explosives, shipping containers. They had already started this, and the company holds those patents for those. So the honeybee population, I'm not entirely sure what are the detection molecules per se, if that's what you're asking. But what we're doing now is that we took some of that knowledge, and they had a patent also for a reader that would read out the sensor output, right? And so they came to Purdue first asking, hey, do you guys make sensors we want to develop these sensors for fish spoilage. You being a food scientist, I'm sure you can appreciate that fish spoils and you know, it's a very expensive food item. And if you can think of it, you know, it's transported all over the world. A piece of salmon, for example, to be brought to North America or wherever it needs to go, it needs to be monitored. And so, you know, the idea was, let's get this fish sensor going. And we were hit at that same time by COVID. And they said, and by the way, if you have anything that looks like a COVID sensor, we should pursue that. At which point I was working already with a professor in materials engineering by the name of Leah Stanshu. And Leah had been working with Richard and myself for some time trying to develop these electrochemical sensors that would recognize pathogens. And through nanotech type of applications, looking at minute differences in the sensor output when there was an interaction with a pathogen. And so we started to go after that and started to develop it because we saw COVID was just there to stay. And we start really working hard towards making the COVID sensors. And the plan there is to, if you think about it, what we want to do is have a handheld device that you can check not only for COVID, but you can check for flu, you can check for other viruses that might come through an upper respiratory sample of saliva. And so we want to build this diagnostic lab for you in your bathroom medicine cabinet so that every morning, if you had something, you could check, or if your child had something, you could check the child. And the ability of having this reader was tremendous because the readout from the sensor gets transmitted from this reader to the cloud, and the interpretation of the test happens on the cloud and is sent back to the person's phone or device. But at the same time, what we can do is use that data to inform the national agencies that are worried about where is the spread, where do we need more help, where are the resources, and so on and so forth. So that's the exciting part. And Purdue is, I gotta say, this university has a lot of great resources that is allowing us to make good progress on the nanotechnology side of things, and in making these sensors, in manufacturing these sensors, looking at processes by which to improve and optimize the sensor performance too. And it's been quite a journey, very exciting so far.
Right.
Yes, Naren, please.
Yeah, thank you. So Thomas, I think one of the, as I mentioned to you before the call, one of the challenges we have in these kind of sessions is it's very difficult for people to ask questions. So how in your education or pedagogy processes, how do you encourage people to ask questions?
Actually, that's a very good question. First, I would say you have to be courageous. Don't, and courageous means that you have to have a certain image of yourself that doesn't matter what anybody thinks or says, you're going to be okay. And I was always the type of student that would ask questions, and I cannot tell you how many times I would have other students come up to me and say, "Oh, I'm so glad you asked that question, because that was something that was also bothering me." So in everything you do in life, there is no risk. There is no risk of asking a question. If people are going to think that you're less intelligent because you asked a question, that's what they think. You are now more empowered with the knowledge by asking a question, even if you think it's not an intelligent question, even if you think, oh, I don't want to speak, I don't want to say something. Many times is better to take the courage and see, oh, I did it and it wasn't so bad and I got my answer. And if you still feel like you're not willing and able or courageous, please type it in. Send me a LinkedIn message, and I'll try to answer your questions as much as I can, as soon as I
can. I think Pratik can ask the question before me. And,
okay, Pratik, please, you can
ask the final
yourself.
question.
Okay.
Hello, Dr. Soers. First of all, boiler up. Just had a quick question. So just wanted to ask, apart from scientific facilities, what other places are close to your heart at Purdue? Thank you.
Oh, okay. Thank you. Yeah, Purdue has a lot of great lecture halls. And one of the programs that I've set up is this lecture hall series where I get my grad students to present to high schoolers. And I get high schoolers to come to Purdue and sit with us in these large lecture halls. I don't know if you know, if you can remember the feeling you had when you first enter a huge lecture hall in an auditorium like that, and how you know, sometimes you feel a little scared. And so I wanted to dispel that. But there's some beautiful lecture halls at Purdue that are really, really neat. And Purdue now too is expanding an area that is very interesting. This Discovery Park has expanded to Discovery Park District, with living and retail space that is starting to come up. And the idea is that we're going to be able to use this to create a smart city like research with living interaction. So there's a lot of, you know, smart city type developments that people want to do. Well, we think we can bring the computation, the engineering, all of the research, the healthy living and climate, and all of the disciplines that are necessary to help build a carbon neutral living space, and do research for sensors inside your living space so that you can reduce the amount of energy, reduce the amount of electricity that you use. So these types of things, automated delivery systems or automated robotic systems that can, you know, pick you up, and you know, this is coming. So we're excited to have this district here in our neighborhood to start to develop some of that as well.
So
There is
I
an
think
airport at Purdue too, I'll say. We have an airport, we have a couple of golf courses. I like golfing.
So I think let's get to know Tommy a little bit more through a rapid fire round that Shruti will host. And then we can, if we have time, we can have another question. I know there's one in the chat box. Otherwise, I'll put Tommy's LinkedIn profile in the chat box and y'all can ask your questions over there.
Great.
Okay, so quick 5 questions. I hope, yeah. So since you've worked in a lot of, like developing a lot of sensors in different domains of science, so which one did you enjoy the most developing it?
Well, so far, this one is the most stressful because we're really pushing this COVID sensor. We're really pushing to get it out there and commercialize it. I'm enjoying it the most. I'm learning the most about it, not only about the science, but also the startup business enterprise and what's required and what does it take. You have to remember, coming from a university, it's very much faculty driven. Here was a company that came to us and is helping to build this other startup. Very interesting.
Okay. So you have developed a lot of transgenic plants, and now there is a trend of organic plants. People mostly prefer having organic. So which one do you support?
Oh, you know, I want to make sure that we are not throwing the baby out with the bathwater, if we say transgenic plants. Many of the plants that are used are or designed were first designed to help the farmers deal with drought, help the farmers deal with cold, or help the farmers produce a more nutritive plant. We have a great example of a vitamin A rice that was, you know, was made, and I'm still very much a proponent of transgenics to improve the value, to improve the way that we are able to grow these plants. There's a lot of people still hungry in this world. And I know organics, I agree with organics if you can, but there's a lot happening in this world that makes me believe that unless we find more sustainable ways that can, if you want organics, okay, but it's not really very scalable if you want, you know. It's difficult for larger companies like McDonald's to deal with 2,000 different farms rather than one or two large farms, if that makes sense.
Okay. What is the one driving force that always motivates you to develop different technology? Just one driving force.
Curiosity.
Okay.
Yep.
Mm-hmm. Okay. When you're not working, what you would like to pursue? Just one thing.
I play a lot of music. I play a lot of guitar. I do sing as well too.
Great. And the final question. So what is your lifelong dream that you always aspire? One dream.
I'm not, I think my lifelong dream is to be able to make a small mark, be able to live a life where I feel like I've contributed whatever I could. I have children, 2, 3 beautiful kids, and my lifelong dream is really for them to be good people, and also to keep up with a little bit of this motivation that I have inside of me to bring fairness to people, and motivate them to do more, motivate them to be better educated. So this is why I've stayed at the university for so long, because I am motivated by that, and motivated by, progress and helping us improve. We take for granted very much that the roads that are paved, that the stop sign, the cars that we drive, and the phones and all of this, that we take it all for granted that this was just made like this when we were born. But no, it took people to do this stuff, and it takes a lot of hard work to make an impact and help a lot of people. So I'm inspired by that, and I hope I can pass that on to them as well.
Thank you so much for answering the question. It was really insightful. Thank you so much.
Thank you for asking.
First of all, thank you so much for this really inspiring talk that you gave. I think the journey that you took from the beginning, and I don't think we talked about your childhood, and I understand you're from Venezuela, or you grew up in Venezuela. Yes. And your journey that you've traversed over these years, from moving from plant biology to cancer biology to entrepreneurship, I think is fascinating. I think more than a question, I will ask the question in more for the other people who are on the call. What does it take to get a postdoctoral fellowship or a graduate program in Purdue University?
So to get a program started, or to get a postdoc? Oh yes.
Or a graduate, yeah.
I really think it takes making contact with people. You can apply. You can apply cold. I'll send my paper and wait. Hopefully, I'll get something. But then you're just somebody else who did the same thing. And I would recommend that if there is research that you like, if there is somebody doing that research at Purdue, please, let's get in touch with them. I can help. As I said, I'm a navigator. I can help with making those contacts. We do have a Purdue India office. If you don't know, we have a lot of great collaborations with different Indian institutions, and it's an easy transition, or it's an easy intake. It does take making contact with people, bridging a connection, creating a bit of a rapport. We will help. If that person wants to bring the person to be a postdoc or to do a PhD in their lab, we will help. And it's a fairly routine thing that we do. We have done this quite a bit. So it's not too difficult, but it does take effort on the individual to make contact.
Thank you so much. I think Charu has put your information on the LinkedIn. So
yeah.
Thank you.
Thank you.
Yeah, I mean, really, thank you so much, Tommy. Every time I talk to you, I'm reminded of just how humble of a person you are. And you have so many achievements, and you're so happy to share the information. And obviously, you started this right at your PhD with developing all of these platforms to educate people. We are indeed very lucky to have been mentored by you today. I feel that now that we're almost at the top of the hour, please send in all your burning questions to Tommy through his LinkedIn. The information I've already put in the chat box, and you can also look up the Purdue India program. If you're interested, there's great agriculture. Purdue is known for agriculture, and there's a great new biophysics structural biology department as well from a professor from Rams who was previously at NCBS. So that's a great program as well. So I'm sorry to everybody who couldn't ask questions, and please send them across, and goodbye for now, and we hope you will tune in next week.
A network should last a lifetime. Let us help you create lasting professional relationships with our world-class mentors through the Biopatrika Industry Mentorship Program. A strategic guidance program unlike no other, full of expert interviews, industry internship opportunities, CV writing, inflection point analysis, life maps, and of course, the gateway to your dream career. For a limited time only, all our services are freely available for you, as we truly want you to succeed.